In the previous class, we had discussed about the biological classification, five kingdom classification. You know about Protista, kingdom Monera, Protista, Mycota, kingdom Plantae or Animalium. So all these five kingdom classifications we have already studied about. Kingdom Monera we did it in the previous class. Today's class we study about kingdom Protista. Prokaryotic organisms were found in Monera. In Protista you have eukaryotic organisms. So what you can notice in this Protista is all of them will be having a nucleus with a nuclear membrane and nucleolus. They will also have double membrane cell organelles like mitochondria, Golgi complex, okay, lysosomes, chloroplasts. All those are found in case of the Protista. So that you have to remember. Most of the majority of the members of kingdom Protista are unicellular eukaryotic organisms, single cell eukaryotes. They have ribosomes of ATS type. You should also understand about that. Then most of them are primarily aquatic organisms. It can be freshwater or marine forms. Okay. Some of these organisms have locomotory organs like flagella or cilia, especially protozoan protists. When we study about them, I will be discussing about that. Then, in case of Monera, you found that they had amitotic cell division or binary fission or budding. You could notice in that Monera. So they did not have sexual and asexual reproduction. They had direct binary fission taking place in bacteria. But that is not the case here. You can notice sexual and asexual reproduction. And you find there is a formation of cells or gametes. Okay, and diffusion of gametes to form zygote. So all those processes are found in the kingdom Protista. But even now kingdom Protista, majority of the organisms are unicellular organisms and eukaryotic in nature. What do you mean by eukaryotic in nature? They have a nuclear wall with nucleolus, that is true type of nucleus is there. They have ribosomes of ATS type. They have double membrane cell organelles. Okay, that is the reason we place them under eukaryotes. Now, kingdom Protista, you can categorize them into the chrysophytes, dinoflagellates, euglenoids, slime molds, and last one is the protozoans. So you can categorize this kingdom Protista into chrysophytes, dinoflagellates, euglenoids, slime molds, and last one is protozoan protists. Slime mold protists, protozoan protists. You have chrysophyte protists, dinoflagellates. So these are the categories under the kingdom Protista. When you study under chrysophytes, you should remember major examples are diatoms. Diatoms and golden algae, which we call it as desmids. They are unicellular. Diatoms are unicellular. You have to remember about them. Okay. Diatoms might be box shaped. We call it as pinnate. The cell wall, they have a, just like your geometry box, they have an outer larger cell wall which we call it as epitheca and a smaller cell wall which we call it as hypotheca. They fit into each other just like your geometry box. Okay, epitheca and hypotheca are the walls that you can notice there, and they have the silica will be distributed in the form of ornamentation. So we call them as frustules. So this is pennate diatom. There is also circular diatoms called as centric diatoms. And the cell walls are embedded with silica. Okay, so that is the reason these they cannot be destroyed very easily. So you have a large number of fossil diatomaceous are accumulated by the dead walls, cell walls of the diatoms. We call them as diatomaceous. From millions of years they have accumulated. So chrysophytes includes what? Diatoms and the golden algae, which we also call it as desmids. The diatoms are called as jewels of the plant kingdom. Okay. So then microscopic and floating passively. So diatoms and desmids are called as planktons. Floating passively and they are not very active. They are floating passively. So we call them as planktons. They are photosynthetic in nature. They have chloroplasts and they undergo photosynthesis. Diatom cell walls, they form a overlapping shells. The outer shell we call it as epitheca, the inner shell we call it as hypotheca. So I have drawn the diagram also here. So walls are indestructible because of silica being embedded in the walls. Today, yesterday's newspaper, if you go through, silica is being tried to be embedded in the tires, rubber tires, so that the tire bursting could be avoided. So silica is going to add to the strength of the organism. Just like silica being embedded in the diatoms, now they are copying from nature even in making of tires. Okay, tires are also being embedded with silica to avoid the tire burst, which leads to lethal accidents. Okay. So remember about this, walls are indestructible because of silica embedding that is found in these diatomaceous wall. So the large amount of cell wall deposits of diatoms and desmides, they are collected for billions of years. Okay, this is referred to as diatomaceous earth. Diatomaceous earth is very much useful. So they are used in polishing, filtration of oils, and also in syrups. They are chief producers in the oceans. Maximum photosynthesis takes place in the oceans, not by the huge trees. Oceans, planktons, they bring about maximum photosynthesis. So the diatoms or the chrysophytes are the chief producers in the oceans. Okay, that is what you have to remember regarding the chrysophytes. Now dinoflagellates, they are the other types of algae you find. Yellow algae, Yellowstone National Park, Xanthophyce is there. Then blue-green algae is in Kingdom Monera, so we can't consider here. Blue colored pigments are there in some of the cells, blue algae, green algae, okay? So red algae, brown algae, they are all based on pigments in the cell. They would be having phycoerythrin, phycocyanin pigments. Based on the types of pigments, the cell colors are there. They might be yellow, green, red, brown, blue, based on the pigments present in the cell. Now the cell wall, if you look into it, they are stiff. They are not made up of silica, rather they are made up of cellulose plates. So the dinoflagellates have cellulose plates. Chrysophytes have walls embedded with silica. Okay. Then they have two flagella. The dinoflagellates have two flagella. One is the longitudinal in position, another is transverse in position. And they are present in the furrows of the cellulose plate. Where are this longitudinal and transverse flagella present? In the furrows of the cellulose plate. You have to remember about. Then red dinoflagellates, Goniolax, example is Goniolax, they are found in the red tides of the ocean. Sometimes you find that some of the tides are reddish in nature. You need not have to get scared. So that is reddish in nature because of the population, overpopulation of this Goniolax, which is a red dinoflagellate. It has multiplied very rapidly, and that is the reason the tides are looking red in color because of the presence of Goniolax, which is a red dinoflagellate. Okay, and this red dinoflagellate, they also release toxic substances, poisonous substances, and they can kill marine organisms like fishes. So they also lead to the death of marine organisms like fishes. Okay, so this is what you have to remember regarding the dinoflagellates. Euglenoids, euglena. Okay, sometimes they are both placed in the plants as well as under protista. So this euglenoids, example euglena you can remember about. So they are freshwater organisms found in stagnant water. Sthiravadantam neer. Stagnant water means something which is not under motion. So it is accumulated water. Okay. So stagnant water you find this euglenoids, euglena. Cell wall is absent in case of euglena, but they have a pellicle. Okay, this pellicle makes the body of the euglena flexible. Instead of cell wall, they have an outermost layer called as pellicle. Okay, they have two flagella, but when you draw the diagram, you show only one. Long flagella is there, another short flagella is there. So euglenoids are photosynthetic in the mode of nutrition because they have chloroplast similar to that of higher plants. Their chloroplast is similar to that of higher plants. In euglena, whenever sunlight is present, they are producers. They synthesize their own food. They are autotrophs. Whenever sunlight is absent, they are heterotrophs. and euglena is a predator. In presence of sunlight, they are autotrophs. They can synthesize their own food. In absence of sunlight, euglena is a predator. It is a heterotroph. Okay, remember about that. Pigments of euglena, that is chlorophyll pigments, they are similar to that of higher plants. That is what you have to remember regarding euglenoids. Example is euglena. Then slime molds. Slime molds, as the name indicates, slimy accumulation of layers. So they are saprophytic protists. They depend upon dead and decaying remnants of plants and animals. So we can consider them as saprophytic protists. They are also found on twigs and leaves, decaying twigs and leaves. You find the saprophytic protists, which are nothing but slime molds. And how they, whenever the conditions are favorable, these slime molds, they accumulate and aggregate to form plasmodium of several feet length. They accumulate and aggregate to form plasmodium of several feet length. They are slimy in nature, slime molds. Okay, so they are found on the twigs and leaves. That also I have given you. They are saprophytic in mode of nutrition. Okay, in favorable conditions, they all aggregate and accumulate to form that group where we call it as plasmodium. Don't get confused with Plasmodium vivax, which causes malaria. This plasmodium is aggregation of all the cells, okay, of the slime molds. We call it as plasmodium, which is of several feet in length. During unfavorable conditions, the slime molds are going to form a thick wall and they form spores. So they are going to form the fruiting body which has on it spores. Spores possess true cell wall which is thick and they help in overcoming unfavorable conditions. See, I was just talking about in unfavorable condition, the slime molds or the aggregate of that body, plasmodium, okay, they are going to develop into fruiting bodies. These fruiting bodies, they bear spores. What are spores? Spores are structures having true cell wall and they are resistant and can survive extreme conditions. High temperatures, high pH, they can overcome that. Okay, so and these spores can be dispersed by air current and they generate, the spores germinate to form a new slime mold at the onset of favorable conditions. So spores are more to overcome unfavorable conditions, unfavorable abiotic factors. Okay. Then protozoans. Protozoans, earlier they were studying it under separate phylum Protozoa in case of some of the classifications of kingdom Animalia. But now they are placed it under protozoans. Most of the protozoans are unicellular and protozoans, according to their, based on their locomotory structures or presence or absence, the classification of protozoans are, you have the amoeboid protozoans, then you have the flagellate protozoans, ciliated protozoans, sporozoan protozoans. So these are the types that you have under the protozoans. So the amoeboid protozoans, you have the example of amoeba. If there is no definite shape, what do you call it as? Amoeboid. Okay, you might have already drawn the diagrams in your records. So amoeba will be producing a false feed. We call it as pseudopodia. Just as if there is a food particle, they produce a false feed and they come together and trap this and they form the food vacuum. Pseudopodia is a false food which helps in the capturing of food. Okay, that is what amoeba has about. And you should also remember about Entamoeba histolytica. They cause amoebiosis in us, human beings. Water infection in Parate, water contamination in Parate rather. So amoebiosis, it is caused by Entamoeba histolytica, wherein they corrode the walls of the small intestine and that leads to dysentery with mucus and further corrosion of the walls by this Entamoeba histolytica. It leads to blood being lost and it might lead to death of an individual also if they are not treated. Okay. Amoebiosis caused by Entamoeba histolytica, by water contamination. Remember about the symptoms of it. So dysentery with mucus or dysentery with mucus and blood. So they are all indications of us being infected by this Entamoeba histolytica. that is nothing but amoebiosis. There are medications which we have to take for that to get the treatment for it. Okay, that is about entamoeba histolytica. Then flagellates. So flagellates, they have flagellum as a locomotor structure. See, the flagellates are free living or parasitic. Okay, they have flagellum as a locomotor structure and they cause sleeping sickness. Trypanosoma gambiensis, sleeping sickness. It is spread from one person to another person through a fly. That fly name is CC. CC. The fly name is CC. Okay, that fly is found only in endemic to African country. You can't say I have sleeping sickness when you are found sleeping in the class. Okay, so unless and until you have visited Gambia or African country and you have been bitten by a CC fly which carried this protozoan, you don't get this sleeping sickness. Okay. So sleeping sickness is also lethal. It might lead to coma and death if it is not treated. So don't be very happy with to have sleeping sickness. It is caused by the flagellate, the protozoan, that is Trypanosoma gambiensis. Gambia is an African country, so they are found there in Gambia. Okay, Trypanosoma gambiensis. Ciliated protozoans, they have cilia. Lot of cilia would be there. Especially, you might have studied about the slipper animacium, isn't it? They have a large nucleus, macronucleus, micronucleus. There will be a gullet like. Then you have tuft of cilia. Have you drawn this diagram in your? Yes. Slipper animacium, paramecium. So they have at the base, they have a tuft of cilia, not French beard. And cilia throughout their body. They have two types of nucleus, macronucleus and micronucleus. Then there will be a gullet-like structure. Okay, so all that you might have studied in your practical classes. So since they have the locomotor structure cilia, we call it as ciliated protozoa. Cilia helps in the paramecium moving very fast. So paramecium is quite fast in moving. So they are aquatic and they have a cavity called as gullet. So you should remember about that in case of this paramecium, which is a ciliated protozoa. Okay, ciliated protozoa. Then the last one is sporozoan protozoa. They are non-motile. Example is Plasmodium vivax, which causes malaria. And again, Plasmodium vivax is the pathogen which causes malaria. So the vector which transfers from an infected person to a healthy person is female Anopheles mosquito. Okay, female Anopheles. So remember about this diseases that are caused by it. There is also one more disease from protozoan, Kala Azar, they call it as Leishmania gonorrhoeae. Okay, black fever, Kala Azar. So that is also one of the protozoan diseases that we should be aware of. So that completes our discussion of kingdom protista.